Let's see now the white blood cells. The white blood cells will make around 1% of the whole blood, and they are important because they are responsible for protection from infections, and they are also responsible for the inflammatory reactions. For the white blood cells, we have two major categories, granular or agranular, and this depends on how they would appear under the microscope. Granular cells would have within the cells some granules, whereas an agranular, they don't have, or these granulars are invisible for us under microscope. For the granular, we have neutrophils, eosinophils, and basophils. For the agranular, we have the lymphocytes and monocytes. Each one of these cells is performing a specific function within our body. Now, most of these white blood cells have very short lifespan, and they are always circulating in the blood, and that circulating level can be immediately raised after infection. Now, let's see the first group, the granular leukocytes. I believe we are going to see some images in the lab. The first group is the neutrophils. This is around 60% of the circulating white blood cells. So this is the highest percentage of white blood cells in our body. And these are the first responders. So whenever there is infection, these are the first ones are going to fight that infection. And usually they are engulfing or eating through phagocytosis these microorganisms. Eosinophil is a relatively low number at normal condition. They are important because they defend against large parasites like worms, and they contribute to some allergic reactions. The third group is the basophil, and these are the lowest percentage of the white blood cells. They contain histamine within themselves, so these cells contain histamine, and histamine is very important in the inflammation and allergic reactions. So these are the basophils. Now for the agranular leukocytes, we have the monocytes. These are around 5%. These are large cells, large white blood cells, and they have the ability to leave the blood, and they transform into macrophages. Macrophages are responsible for detecting infections and microorganisms within the tissue. And the last group is the lymphocytes. Lymphocytes are around 30%. This is the second large group after the neutrophils. These are also circulating white blood cells, and they are important in immune response. Think of viral infections, cancer, all such type of diseases. Lymphocytes are responsible for dealing with them, and we have two types, B lymphocytes and T lymphocytes. The other group is of formed elements is called platelets. These are not cells. These are fragments of cells produced by cells called megakaryocytes in bone marrow. These cells would break into fragments, and these fragments are the platelets. They are essential for blood clotting. If blood vessels is injured, platelets will initiate the clotting process. Also, they would participate in the repair process. So these are the platelets. Now, with them, they might cause some clotting disorders like bleeding disorder, like something called hemophilia. This is deficiency of one or more clotting proteins. In this case, the blood cannot clot, and if there is an injury, the blood would continue to run out of that cut till something, been taking, like, sealing stitching the cut or something like that to stop the bleeding. Or if you leave it by its own, it will take so long time to heal. Some drugs, some medication will interfere with the homeostasis of clotting, like aspirin. Aspirin blocks platelet clumping and delays the clotting. Now, let's talk about the human blood types. Human blood types is important to determine the blood transfusion. Blood transfusion is administration of blood directly into bloodstream of another person. And success is dependent on matching the blood type of the donor with that of the recipient. We have 4 groups of blood type, A, B, AB, and O. And we have the Rh positive and Rh negative. And I believe you understand there is A plus, A minus, B plus, B minus, and so on. Now, in order to understand the human blood types, we need to understand 2 things, the antigens and antibodies. And I will move to OneNote in order to be able to explain them in a better way. Now, let's talk about the antigen. Oops, we need this. Antigen. I want to reduce the size. Maybe this. Okay. Now, antigen, think of them as the ID, identifiers. Now, for everything within our body, like cells or otherwise, they have their IDs. This is a cell, and the antigen, the antigen would be displayed on the surface of the cell. Okay? Now, if this is like a cell, this is a cell from the body. So these are self antigen. Think of if you are working in a company and that company is giving you like an ID card, okay, with some names on it. That is the self identifier. For the cells, this is the self antigen. Now, let's assume there is an external cell, like a cell or a virus or microorganism, invading the bloodstream. This microorganism also has IDs on it. These antigens are not self, meaning they do not belong to the body. So in this case, they are foreign antigen. Okay, foreign antigen. So when there are the white blood cells, let's draw them in purple, they will come and examine these antigens. They will recognize that this organism does not belong to the body, and they will start to attack it. It's exactly like you are in a company and you were asked by a security guy to present your ID, and you present your fake ID. So the security guy would know that this is fake and will arrest you, okay, or take you out. So this is the concept of the antigen. Antigens are identifiers. They are displayed on the surface of cells, viruses, microorganisms, etc. Anything that comes into our body has this ID. Now you might wonder, what if it doesn't contain any ID? In this case, the white blood cells cannot see it, and that is extremely dangerous and rare, by the way. Okay, so these are the antigens. That's the first concept. The other concept we need to work on is something called the antibody. Antibodies, think of them as arms. Okay? Arms released by cells to detect the antigens. Okay? Arms, you can say scanners. You know, when you go to Walmart, for example, to buy something, they have these, tools to scan the barcodes, right? So these are the antibodies. They are swimming in the bloodstream, scanning these antigens. And if they find or detect a non-self antigen, they will bind it to it. They will bind to it, meaning that they will catch it and prevent it from going anywhere further in the bloodstream. Usually, these antibodies are produced by the white blood cells. White blood cells, they release these antibodies in a huge amount, and these antibodies will circulate into the blood, catching any foreign antigens. Okay, this is very brief, explanation of the antigen and the antibody. Now, to understand the white blood cells, let's start a new page. To understand the white blood cells or the red blood cells types. So this is blood grouping. Red blood cells, they have self antigens on their surface. These self antigens can be in form of two shapes: circular or rectangle. If it is rectangle, we'll call it A. If it is circular, we'll call it B. I'm not sure why this keeps moving like this. Sometimes the red blood cell would display both. And in this case, it will be A, B. Sometimes it doesn't display anything, and that is the blood group O. Okay, so this is the blood grouping. If blood cell has antigen A, then it is A. If it has B, then it is B. If it has AB, then it is AB, and if it has nothing, it is O. Okay, so this is the blood grouping. Let's go back to the lecture. Okay, so as you can see here, these are antibodies This is antibody, antibody, antibody. This is a self red blood cell, self red blood cell. This is a foreign red blood cell. These antibodies are going to catch it and preventing it from circulating further into the blood. So all of this we explained. Yeah, we need to go back and explain this concept. That individuals have antibodies against the antigens, not on their own red blood cells. What does that mean? Now let's assume that there is a person and that person has the blood group A. Okay, this is blood group A. We said the antibodies are responsible for attacking foreign objects. Now, I don't like to use the word foreign because of the current political atmosphere in US, but this is the word we are stuck with. Okay, antibodies, as we said, back to the topic, back to science. Antibodies are attacking, or their function is to attack foreign objects. Now, do you think that the A red blood cells in this case are foreign objects? Of course, no. This is their blood group. So do you think that this person has antibodies that are going to attack the A? So I would write the question. Does this person have antibody that is going to attack blood group red blood cells A? Of course, no. Why? Because red blood cells A are self cells. Okay, now what is the other red blood cell type? We said we have the red blood cell B. The red blood cell B, they have this circular antigen. Now, do you think a red blood cell B, red blood cell B here is a self? So this is B. So the question is, this is 1. 2. Do you think that red blood cell B is a self cell? No. Right? So the third question, does it make sense, or would the body attack it? Yes, through antibody. So now this person has an antibody that is specific for the B, and these antibodies are going to attack the antigen B. So these antibodies are called anti-B antibodies. Anti-B antibodies. This person does not have the anti-B A antibodies. He has the anti-B antibodies. So for these antibodies, usually a human body will have the antibodies of the blood group that is different from its own. So human body would have antibodies for the blood group that is different than its own. In this case, this human body or this person has anti-B antibodies because his blood group or her blood group is A. Imagine if they have the anti-A in this case. Then these antibodies will attack the cell, destroying it, and eventually will kill that person, which is something we don't want it to happen. So that's why we don't have the anti-A antibodies. Okay? Let's go back to here. So if the recipient of a blood transfusion has antibodies against the donated cells, a severe, possibly fatal reaction may occur. Again, if you are giving someone with the blood group A, okay, if you are giving him a B, his body has anti-B. So, and if you introduce the B cells, then there will be this transfusion reaction. So this table summarizes the different types of blood. And as you can see, type A has antigen A, type B has antigen B, type AB has both, type O has nothing. Now in the plasma antibodies, we said we are going to have the opposite one. For type A, we have the antibodies B. For type B, we have the antibodies A. For type AB, there is no antibodies. Otherwise, both will be reacted or attacked. And for the type O, since we have no antigens here, then we will have both antibodies. And this is the incidence of distribution between the different types of blood group against different ethnicities. Now, the ABO blood typing is based on the A and B antigens. As we said, this is like a summarize for what we talked about. Then we did not talk about the RH because it's the same concept. Now, imagine that is the A plus. Then in this case, it will have the A and it will have something called the D. So this is responsible for the A, this is the RH, okay? In this case, we will refer it to positive. If it has only the A, then in this case, it will be A negative, since RH is missing. The same concept. And again, these people, they have, for example, the antibody B or the anti-B antibody. These will have anti-P and anti-RH, since they are missing the RH antibody. These, they are not missing it, so we don't need that antibody. Now, why this important? Why the RH important? Because this might result, especially in pregnant women, in something called hemolytic disease of newborn RH positive babies. Especially this happen if there is a woman that she is RH negative, but her baby is RH positive. What will happen is that when, as you can see, RH negative woman with RH positive baby, that some of the fetal red blood cells, some of the baby's red blood cell would transfer back to the mother, and the mother is Rh negative. Now we have these Rh positive cells. Now for the mother, yes, these are from her baby, but for the body, these are foreign objects. So her body will react by producing antibodies against the Rh, against the positive cells. Now for the first pregnancy, there is no problem in this. But in case there is a second pregnancy with Rh positive, these antibodies from the mom would move to the baby and attack her red blood cells. This unfortunately would kill the baby. This unfortunately would kill the baby and would lead to miscarriage. So usually what's happened, if there is an Rh mom, she would receive a shot to prevent formation of these antibodies, and in this case to make the baby survive. This is animation about the blood type. Please watch it. Now, in order to make sure that we have a correct blood typing, we need to do a cross matching, and this is something we are going to do in the lab. So in order to do this, we would mix the blood with solution that has either anti-A or anti-P antibodies. And look for the clottings. Okay, let's go back to one note. So we said we have a solution, or let's start with the blood. This is the blood, okay? And this is the solution. This solution has anti-A antibody. This one has the anti-P antibody. Let's zoom in to see what does that mean. If I zoomed in, this is the anti-A antibodies. They will appear like this. Let's assume. And this is the anti-B antibody. Okay? Now, let's see the blood. If we zoom into the blood, we have red blood cells with antigen service, with antigen on their surfaces. So assume that this is the antigen, and the same situation is here. This is the same blood, okay? And this is the antigen. Now, when we mix them, what are we going to have? Let's draw it in a mid color between them. We will have the red blood cells with their antigens and the antibodies binding to them, right? Because these are anti-A antibodies. Here we have the same cells but the antibodies cannot bind because these antibodies, they have different shape. What we will see eventually, that in this group we will have clots of blood, whereas in this group we will have no clots. Now, clotting means that the antibodies found antigens. And since we know that the antibodies are for A, then the antigens are for A, then the blood group is A. Here, antibodies B couldn't bind, so no antigens B. In this group, in this case, the blood group is A. And as you can see here, when we have clots in both, then this is the AB. Lastly, some disorders in the blood. We have the anemia, the reduction in oxygen carrying capacity due to different reasons. Iron deficiency, hemogric anemia, pernicious anemia, hemolytic anemia. Each one of these has a different reason. Also, we have the uncontrolled production of white blood cells. That's leukemia. This is the blood cancer. And we have different types depending on the type of the cells, of the white blood cells being produced. We have leukemia, we have multiple myeloma. And I believe that would end this chapter. Please post your questions, your videos, your thoughts into the forum, and we will talk more about this in the lab.